Alright, going back to this pathway, we have covered glycolysis of glucose and other 6-carbon sugars, homolactic fermentation, gluconeogenesis, and the regulation of all these processes. With that in mind, we are now ready to talk about glycogen, which as a molecule serves the essential purpose of storing glucose. As a brief overview of what we will discuss, I want to start by introducing some important terminology on the structure of glycogen, then go over the mechanism of glycogen synthesis and glycogen breakdown, and then finish with the regulation of this entire process. I want to mention right away though that in this section I won't discuss just yet how the metabolism of glycogen is integrated through different organs, as we will cover this topic a little later. I will talk a little bit about the hormones when we get to the regulation, but for the moment the aim is really to focus on the reactions. Before we get to them though, let's consider what glycogen generally looks like by considering a short portion of this molecule. As you can see, glycogen is a polymer of glucose monomers, or in other words, a molecule made out of glucose units, all branched to one another. Now, because of this branched, chain-like structure of glycogen, it generally has two types of extremities: many non-reducing ends, here shown in blue, and one reducing end, shown in green. The terminology for reducing and non-reducing ends has to do with some organic chemistry concepts that are not too relevant for us. Here, really the most important point to keep in mind with these ends is that all the synthesis and breakdown reactions will occur at the non-reducing ends. In terms of the branching, the glucose units within the glycogen molecule can form two types of bonds. First, we have the bond between carbons 1 and 4 that allow the units to be linked in a chain together, here shown in red, and second, every 8 to 14 glucose residues, a bond between carbons 1 and 6 that allow the glucose chain to branch out. This ability to form multiple branches is crucial for the normal health of a cell because it allows to increase the number of non-reducing ends, which again are the sites of breakdown and synthesis. Lastly, you will notice that the glycogen molecule here begins with glycogenin, which is a very important enzyme that allows glycogen molecules to form in the first place. It turns out that the enzymes that we will see shortly that mediate the breakdown and synthesis of glycogen can only operate on already established chains, and thus, glycogenin is crucial to get everything started. To do so, this enzyme is a glycosyl transferase, which means that it has the ability to move around some glucose molecules. With this ability, glycogenin will take up to 7 glucose units and add them to its structure on its tyrosine 195, after which the other enzymes will be able to take over the process of elongation, branching, and breakdown. You will notice as well that the glycogenin covalently binds the reducing end. Now, to get a more macroscopic idea of what a glycogen molecule looks like, you can consider this schematic with the many non-reducing ends in blue and branching points in purple. At this scale, these glycogen molecules are also known as beta particles, and many beta particles can make up an alpha particle, which you can see on this electron microscopy image. The alpha particles are what will typically be observed in living tissues, although this image simply shows isolated glycogen. With this being said, we can now consider how glycogen is synthesized and broken down, starting from glucose. For this process, we will assume that glycogenin already has a few added glucose residues, and the other enzymes that I will shortly introduce are able to work. Also, given that the glycogen molecule takes quite a lot of space, we will simply consider a given amount of glycogen and this same amount plus 1 unit of glucose to illustrate the transitions between adding and removing one glucose residue. The blue R in both cases represents the rest of the glycogen molecule. Now, if you recall our discussion on the principles of metabolism, you might remember that glycogen synthesis is an anabolic process, which implies that it will require an input of energy to happen. This input of energy is very important because there is no reaction that will be able to take glucose and add it directly onto glycogen in a thermodynamically favorable manner. Instead, the glucose will have to be charged with energy first to make the process spontaneous. To obtain this charged version of glucose, glucose first gets phosphorylated by exokinase to make glucose 6-phosphate. Then, this glucose 6-phosphate gets reversibly mutated to glucose 1-phosphate by phosphoglucomutase. As of yet, the glucose that we have in the form of G1P has some energy in the phosphate, but not enough to be integrated into the glycogen. It is the enzyme UDP glucose pyrophosphorylase that generates the charged glucose called UDP glucose by adding UTP to G1P. This reaction takes the UMP portion of UTP and leaves two phosphates bound together. At this stage, this reaction is not very spontaneous and can go in either direction, but the cleavage of the two phosphates by the inorganic pyrophosphatase releases a lot of energy that fuels the making of UDP glucose. This detail is very important because if you imagine a cell with a lot of glucose that it wants to store as glycogen, the cell does not want to wait a long time for UDP glucose to be made, so accelerating this reaction with the cleavage of two phosphates makes this entire thing efficient, which is most of the time what the cells aim to do. Now, the newly made UDP glucose can be used by the enzyme glycogen synthase and be integrated into glycogen. Obviously, the glucose units in glycogen do not bear a UDP, and as such, it gets released during the reaction. Also, keep in mind that here, the newly added glucose binds to the fourth carbon to elongate the chain. The branching at the sixth carbon occurs via a different mechanism that we will cover shortly. A final reaction that I want to mention when it comes to the synthesis aspect of glycogen is that UTP can be recycled by a reaction catalyzed by nucleoside diphosphate kinase. As you can see, this reaction takes ATP and UDP and swaps a phosphate group to produce ADP and UTP. That UTP can then return and be used to make UDP glucose again. So, when we consider the energetic balance sheet of this entire process, it essentially costs 2 ATP to add 1 glucose molecule to glycogen. 1 ATP comes from the hexokinase reaction, and the other from the recycling of UTP. Now, moving on to the breakdown of glycogen, with what we have established here, the process is pretty straightforward. The enzyme glycogen phosphorylase uses 1 phosphate molecule to cleave the chain and produce a G1P molecule, which brings us back at the starting point of this entire pathway. Since phosphoglucomutase is a freely reversible enzyme, G1P can be converted back to make G6P. In comparison to the synthesis aspect of glycogen, which happens in conditions of high energy and high amounts of glucose, the purpose of the G6P produced from glycogen breakdown will be dependent on the tissue and its needs. The main reason for that is because G6P is at the crossroads of many pathways, namely glycolysis and gluconeogenesis. So, for example, if we consider the muscles when someone is exercising, the G6P will be able to go through the glycolysis route, produce 2-pyruvate and some ATP to fuel muscle contraction. On the other hand, if we consider the liver under fasted conditions, the G6P can go the gluconeogenesis route and produce glucose to be released into the bloodstream. Recall that the enzyme that allows the conversion of G6P to glucose is located selectively in the organs that perform gluconeogenesis, so the liver and the kidneys. Hopefully, at this stage, you can start to see how some of these pathways that we have covered overlap and coordinate with each other to support the health of the organism. Obviously, these changes do not happen on their own and often require the assistance of hormones to control the fates of the metabolites, and we will cover shortly the regulation that makes all of this possible. Now, what I want to cover before we discuss regulation are the general rules that govern how glycogen is synthesized and broken down. To do so, let's start from the very beginning with glycogenin. As I mentioned before, glycogenin will kickstart the glycogen molecule by creating a primer of about 7 glucose residues. Recall that the very first glucose unit that is covalently bound to the tyrosine 195 will be the only unit with a reducing end for the entirety of this molecule. When the primer is complete, glycogen synthase can now take over and start elongating the chain at the non-reducing ends. Obviously, as we talked about, for glycogen to be efficient in storing and delivering glucose to the cell, it's better for its structure to be branched. This is where the branching enzyme comes into play. Basically, what this enzyme does is that it takes a chain of 7 glucose residues and binds it between carbons 1 and 6 to create a new branch. From there, glycogen synthase can elongate the chain again. There are 2 important rules that govern the branching. First, the segments that the branching enzyme takes must come from a chain that is at least 11 glucose units long. So here, for example, since the chain is 14 glucose units long, the last 7 units can be taken to make a branch. If the original chain elongates and a new branch is made, The second rule is that the branch must be at least 4 residues away from an already existing branch. Now, when it comes to the breakdown of glycogen, let's consider the rules on another branch. So, when the cell wants to break down glycogen, the vast majority of the glucose will be provided by glycogen phosphorylase, which will go at non-reducing ends and cleave the units via phosphorylysis to produce G1P units. One caveat with glycogen phosphorylase, however, is that it will only operate until it reaches the 4 last glucose residues of a branch. To completely remove a branch, it is the debranching enzyme that will complete the job. To do so, the enzyme has 2 activities. First, it will take the 3 glucose units that are at the edge of the chain and displace them on a different branch. Secondly, for the remaining unit, it will cleave it through a hydrolysis reaction and directly produce glucose instead of G1P. This aspect between the debranching enzyme and glycogen phosphorylase is pretty important, as about 10% of the glycogen is converted to glucose directly and the remaining 90% to G1P. With this being said, these 5 enzymes are the ones mostly responsible for shaping glycogen. Now, if we consider back our diagram for the synthesis and breakdown of glycogen, you will notice that with the glycogen synthase and glycogen phosphorylase reactions, we have a substrate cycle. Recall that substrate cycles happen when we have 2 opposing irreversible reactions producing the reactants and products. The most important point with these cycles is that since both reactions are irreversible and can happen spontaneously, they must be regulated to have a committed direction. This is where, if we consider the metabolic map for a moment, you will see that glycogen synthase and glycogen phosphorylase are both a part of a complex network of stimulatory and inhibitory signals. In the same fashion as the enzymes involved in glycolysis or gluconeogenesis, the 2 main ways to regulate the activity of these proteins that we will cover is via allosteric and covalent modifications. One thing I want to mention though is that in comparison to allosteric modulators, covalent modifications are a bit more tricky to cover because, as you can see from the metabolic map, they are intrinsically linked with the pathways of hormones. So as we did with the other sections, I will provide an overview for the hormones and explain how they relate to glycogen with their key downstream effects, but we will cover in more detail in the later section the organs responsible for releasing them and how they impact as a whole the metabolism of different tissues. Now, to cover the different regulatory mechanisms, I want to first simplify the reactions we have covered to have some space to work with. So, as a quick reminder, everything starts with glucose 6-phosphate, or simply G6P. This G6P can be converted to G1P via phosphoglucomutase, and the enzyme UDP glucose pyrophosphatase can produce UDP glucose out of this G1P. Then, the enzyme glycogen synthase can integrate the UDP glucose to elongate glycogen. On the other hand, the enzyme glycogen phosphorylase can cleave the glycogen to get back one G1P unit. So, as we will go along through this discussion about regulation, you will see that there are a ton of different players involved in regulating either glycogen phosphorylase or glycogen synthase. Yet, regardless of the mechanism, all the regulatory signals will contribute in promoting either 2 scenarios. The first scenario is that the cell has an excess of glucose and energy and wants to store it in glycogen. For this to happen, glycogen phosphorylase must get inhibited and glycogen synthase must get activated. The other scenario is that the cell needs glucose, so it will break glycogen to get some. In that case, glycogen phosphorylase must be activated and glycogen synthase inhibited. This scenario, as we said, is a little bit more complex because the glucose made from glycogen can be used for gluconeogenesis in anabolic tissue such as the liver, whereas in muscles, the glucose will be used for glycolysis to make energy. Another important point is that for each scenario, there are dedicated hormones that will coordinate a lot of the regulation. In the first, the prominent hormone that stimulates glycogen synthesis is insulin. In the second, the hormones epinephrine and glucagon are heavily responsible for glycogen breakdown. With this in mind, let's start by considering factors that promote the second scenario. As we have seen with glycolysis and gluconeogenesis, an important regulator for all these processes is energy itself. An important signal to indicate that energy might be running low is the accumulation of AMP, which is simply ATP with 2 less phosphates. As you might expect, AMP, which again signals for low energy, is an allosteric stimulator of glycogen phosphorylase, and this makes sense as the reaction catalyzed by glycogen phosphorylase will produce glucose to make energy. Now, the rest of the activation that glycogen phosphorylase receives comes primarily in the form of covalent modifications that are directly triggered by hormones. As such, we need to briefly discuss the intracellular mechanisms that these hormones initiate. Fortunately for us, glucagon and epinephrine, which are both responsible for breaking down glycogen, activate the same pathway, but just in different types of tissues. Glucagon signals to the liver to release glucose in the bloodstream, so glycogen breakdown leads to gluconeogenesis. Epinephrine, on the other hand, signals for muscles to use the glucose to get energy, so the broken down glucose is processed through glycolysis. For these two hormones, the pathway in question is the metabotropic GS pathway, and it is activated by glucagon binding to its glucagon receptor or epinephrine binding to its beta-adrenergic receptor. If you need a more in-depth explanation on this pathway, you can consider this video that I've made on the subject, but what is important for us with respect to this is that the binding of either glucagon or epinephrine causes the downstream activation of protein kinase A, or simply pKa. By itself, pKa is a very important protein that has a ton of different targets inside cells, but in terms of glycogen, pKa phosphorylates and activates a kinase by the name of phosphorylase kinase. When active, phosphorylase kinase also activates glycogen phosphorylase by phosphorylating it. Altogether, this promotes the breakdown of glycogen. In addition to that, pKa and phosphorylase kinase also phosphorylate glycogen synthase, but in this case, instead of getting stimulated, this causes its inhibition. This is our first example of a dual effect in this pathway, and it is a very important mechanism to ensure that cells completely commit to a given pathway. Now, in addition to phosphorylase kinase and pKa, there are also two other kinases that are responsible for inhibiting glycogen synthase, namely glycogen synthase kinase 3 beta and protein kinase C. We will see shortly how glycogen synthase kinase 3 further connects to this pathway, but in terms of PKC, this is a kinase that is also triggered by epinephrine. However, it is not triggered by the beta-adrenergic receptor, but rather the alpha-adrenergic receptor, and thus activates the metabotropic GQ pathway. Again, if you want more information on this, you can consult this video where I discuss the GQ mechanism in more detail. An important detail to note with this pathway is that it leads to a rise in intracellular calcium, and calcium is a very important stimulator of phosphorylase kinase, which again emphasizes the commitment to glycogen breakdown. Another reason why calcium is relevant to point out is because muscle contractions also cause the increase in calcium, and this makes sense as muscles need energy to contract, so having calcium stimulate phosphorylase kinase will contribute in getting more glucose to supply the energy via glycolysis. In terms of the first scenario, these are the main signals that contribute to breaking down glycogen. As you can see, although there are different mechanisms at play, all the regulators that contribute to glycogen breakdown stimulate glycogen phosphorylase and inhibit glycogen synthase. Now, for the second scenario, which is to synthesize glucose, we can begin by considering some of the allosteric control that is dedicated to accomplish this task. Here again, energy plays an important role as ATP inhibits glycogen phosphorylase. Given the AMP activation, it should be intuitive why high levels of ATP promote glycogen synthesis by inhibiting glycogen phosphorylase. In addition to energy, glycogen synthesis is also promoted by high levels of glucose and G6P. In the liver, high amounts of glucose contribute to inhibiting glycogen phosphorylase. For G6P, high levels of it have a dual effect of inhibiting glycogen phosphorylase and stimulating glycogen synthase. This allosteric regulation should also be intuitive because if high amounts of glucose and G6P accumulate, then it likely means that energy is plentiful and there is no need for glycolysis, so the extra glucose and G6P should be stored into glycogen. Now, when it comes to the covalent regulation of glycogen synthesis, the main actor that contributes to promoting the synthesis is phosphoprotein phosphatase 1, or simply PP1. To avoid any confusion on how this protein operates with the rest of the pathway, I want to briefly discuss the difference between a kinase and a phosphatase. Very simply, kinases are proteins that use ATP to phosphorylate other proteins on their serine, threonine, or tyrosine residues. Phosphatases, on the other hand, are proteins that remove this phosphorylation and make the proteins go back to their dephosphorylated state. The very important thing to know about this process is that both phosphorylation and dephosphorylation can activate or inhibit a given protein. This is crucial to keep in mind to avoid falling in the trap of associating phosphorylation with activation. As such, when we consider the diagram again, phosphorylation of glycogen phosphorylase stimulates it, but phosphorylation of glycogen synthase inhibits it. Because of that, PP1 dephosphorylates glycogen synthase and thereby activates it. Additionally, PP1 dephosphorylates phosphorylase kinase and glycogen phosphorylase, which inhibits these proteins and allows the cell to double down on glycogen synthesis. The PP1 protein itself gets regulated by the PP1 inhibitor. As you might expect, this inhibitor gets activated by the glycogen breakdown side, and it happens via phosphorylation by pKa. This additional phosphorylation by pKa ensures that PP1 stays blocked so that there is no competition during glycogen breakdown. However, when PP1 is active, another one of its targets is to dephosphorylate the PP1 inhibitor. So, as you can see from this diagram, basically everything that gets activated or inhibited by phosphorylation, PP1 is there to undo it and cause the opposite. In terms of hormonal regulation for glycogen synthesis, as we said, the main hormone responsible for that is insulin. With respect to metabolism, insulin is a very important signal to remove glucose from the blood, and one good way to go about it is to promote cells to store glucose as much as possible so they can intake more and remove it from the bloodstream. The intracellular pathway that insulin triggers is quite different from glucagon and epinephrine, as insulin activates a tyrosine kinase receptor and not a G-protein coupled receptor. In the cell, insulin has many effects, but the two notable actions from this pathway that are relevant for us are that insulin inhibits glycogen synthase kinase 3 and stimulates PP1. Altogether, these actions promote the dephosphorylation of glycogen synthase and the synthesis of glycogen. A final detail that I want to add to this diagram is that when G6P stimulates glycogen synthase, it does so by facilitating its dephosphorylation. So, in all, you can see that the regulation of glycogen is pretty complex and involves many actors. If you keep in mind that all these signals contribute to either two scenarios, I think that may help getting better intuition on this process. One limitation, unfortunately, with the way this diagram is set up is that the stimulation and inhibition arrows do not differentiate on the mechanism of regulation. What I mean by that is that, for example, AMP and phosphorylase kinase both have green arrows pointing at glycogen phosphorylase, but the two regulators use different mechanisms to do so. One is an allosteric control and the other is a covalent control. Because of that, it's important to understand the meaning of the names, for example kinase and phosphatase, to be able to deduce their mechanism. With this being said, this concludes our discussion on glycogen metabolism and overall discussion on carbohydrate metabolism. In it, we covered glycolysis, gluconeogenesis, homolactic fermentation, glycogen synthesis and breakdown, as well as the regulation of all these processes. Also, you can see on the left the extent of the metabolic map that we covered with these topics. When we consider the regulatory mechanisms of each of these pathways, you will notice that hormones are involved in all the regulatory steps. So, just as a quick note for later, when we will discuss hormones in more detail and their integration in the human body, we will revisit these regulatory steps to really understand how these pathways all work together. Alright, with this section on carbohydrates now covered, I want to consider how energy is produced in the mitochondria. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.